Okay. So this is Pittsburgh, just in case you were curious. It's not the smoky town it was 50 years ago, and certainly not when Mark Twain called it hell on earth. We're a very clean city now. So what I'm going to be talking about is schizophrenia. Now, schizophrenia is a really complex disorder. It's a thought disorder comprised of three major symptom classes. The positive symptoms, they're called positive symptoms because they're things that are added on to a normal personality, like hallucinations and delusions. There are negative symptoms, which are things that are missing from a normal personality, like social withdrawal, inability to maintain a job or relationships, and cognitive symptoms. Now, this is a drawing that's made by a schizophrenia patient. Now, they don't see things the way that we do. So these are drawings of a cat that's, of course, very elaborate, but also very strange and provoking kind of uneasiness because of the bizarre nature of this. As far as the positive symptoms of schizophrenia, the primary symptom that they tend to show are these auditory hallucinations. Now, this is an actual email that I received. I have no idea whether this person was diagnosed with schizophrenia, but it's classic schizophrenic thinking. What he writes is, I have a problem with the neuroradio implanted in my brain, and I'm looking for a neurologist or radiologist who's removed or diagnosed a radio chip circuit in human brain. Can you please let me know if you have diagnosed or removed a neuroradio, and if you have any knowledge of someone who has, and how much does the removal of the radio cost? The arguments of advantages of having one in the brain, I'm not quite familiar with, do not convince me, etc. They hear these voices, and they often come up with these magical explanations of where they're coming from, that these are thoughts being broadcasted in their head, or somebody put a radio receiver in their head, or they're getting messages from between channels on a television set. They also show, again, negative symptoms, these problems with social interactions, and also a lot of cognitive deficits, especially in the way that they express themselves in speech. So this is an example of a writing of a schizophrenia patient. What they say is, the sea beach gathering homestead building upon the site of the bare mountains, time placed to the dunce, to the recovery of the setting sun, upon the stream, poling paddle boat, Mickey, Rooney, Bill, proceeded of to the enlivenment. No matter how many times you read this, you're not going to come up with a deeper meaning. This is this classic schizophrenic word salad, where words seem to be mixed in, where there may be relationships between words, but the logical flow of thought is completely absent. Now, how can we study something as uniquely complex and cognitive in humans as schizophrenia in a rat? Well, we don't know whether we can really make rats schizophrenics. We don't know whether we can get them to hear squeaks, and it's really hard to get them to lie down on a couch and tell us about their mothers. But what we can do is to look at a number of things that are seen in human schizophrenia patients, both epidemiologically, with imaging studies, with pharmacological responses, and then try to translate them to the rat and back and forth. In doing this, we've actually gotten a lot of advances. Now, one thing that we've used is a mitotoxin, methylazoxymethyl acetate, or MAM. In fact, I think you've seen a poster and a presentation on this already. MAM is a DNA alkylating agent. Now, schizophrenia is genetically linked. It's not genetically determined. It's not like Huntington's, where if you have the gene, you get the disease. But genes confer risk factors. But also stressors that occur during gestation, especially during the second trimester, are associated with higher incidence of schizophrenic births, like severe influenza infections, higher incidence of schizophrenic births in the spring, which correspond to the second trimester being over the winter, things along those lines. So what we do is we give this DNA alkylating agent at gestational day 17, which is about equivalent to the rat's second trimester, and then look at these animals as adults. And what we find when we do this are a number of things that are consistent with schizophrenia in humans. First, if you look at the anatomy, what you find is the limbic cortical structures are thinner, not dramatically, maybe 10 to 20%, but it's the same structures that you see thinned in the human schizophrenic cortex post-mortem. If you do stereology on these, what you find is that you don't lose a lot of neurons, but the neurons are compressed closer together. So you're losing neuropillar complexity without losing a lot of neurons. But you do tend to lose a specific type of neuron, and these are the ones containing GABA and parvalbumin markers, especially in the frontal cortex and hippocampus. We see it in our MAM rats, you see it in schizophrenia patients. We see a lot of deficits with respect to behavior. There are some tasks that they do fine. There are some tasks they do better than normals. But what they do show are deficits in sensory gating, like pre-pulse inhibition of startle, deficits in executive function, like reversal learning and extra-dimensional shift, and deficits in the way that they deal with emotional stimuli, like latent inhibition and social interaction. And then finally, they show differences in pharmacological responses. In some studies that were done, So if we do a Y-maze alternation task, where there's a Y-maze where there's one arm with the flashing light and one arm with the steady light, and they have to learn to go towards the flashing light. They learn that discrimination significantly faster than controls. But when we do the reversal, they never learn the reversal. And we think that they learn the initial discrimination for the same reason they never learn the reversal, and that once they make the correct choice, they never test the alternate choice. And they also show differences with respect to pharmacological responses. For example, in some studies that were done back in the 50s before ethics were invented, they showed that if you give schizophrenic patients PCP, you get a dramatic relapse to their schizophrenic state that's it's a condition that's indistinguishable from a relapse and can last for weeks. And what we see in the MAM rats is they're much more sensitive to PCP and show a much greater locomotor response to amphetamine, but they only show it in the adult. They don't show it pre-pubertally, which is interesting because schizophrenics don't show these deficits before puberty. These are things that emerge in late adolescence or early adulthood. Mm-hmm.
I have a question about the dopamine expression. Did you observe the differences between the dorsal part of hippocampus and ventral, or in this model?
In this model?
Yeah.
So
is the expression the same in dorsal and ventral hippocampus, or maybe there are differences?
With respect to parvalbumin?
Yeah.
We see the biggest effect in the ventral hippocampus. We see some effect in the dorsal hippocampus, but the only big significant changes are in the ventral part, especially in the subiculum, which is...
Do you know any data, pharmacological data, that maybe some compounds will be effective in reversing this effect?
Yes, and I'll get to that. Now, the increased amphetamine response is consistent with the role for dopamine in schizophrenia. And there's a long history of a dopaminergic involvement in schizophrenia. All antipsychotic drugs that are in use today are primarily dopamine antagonists. So they work on the D2 receptors. There are some that, of course, have affinities for serotonin, some for acetylcholine, some for norepinephrine, but they're all given at doses that produce a certain level of blockade at D2 receptors, independent of their off-target effects. Drugs that increase dopamine release, like amphetamine and L-DOPA, will worsen the positive psychotic symptoms of schizophrenia patients, and if are given at high enough doses to controls, will mimic a psychotic state. And imaging studies show a big increase in amphetamine-induced dopamine release. Now again, this is done with raclopride displacement. So you give tracer doses of radio-labeled raclopride, give a low dose of amphetamine, and look at raclopride displacement as an indice of dopamine release. And what was found by Anissa Abidargham and Mark Laurel is a significantly greater raclopride displacement in schizophrenia patients that's proportional to the ability of the amphetamine to worsen the psychosis. So we have this identity between overstimulation of the dopaminergic system and positive symptoms. The problem is no matter how much people have looked at the dopamine system itself, there aren't dramatic differences between schizophrenics and normals. Instead, what we're starting to think is the dopamine system itself is probably normal, but it's being dysregulated by other structures. And one that's gotten a lot of attention is the hippocampus. So in the MAM model of schizophrenia, there's a hyperresponsivity in the dopamine system that's at least analogous to what you see in schizophrenia patients. And if you look at metabolic imaging studies, fMRI, what you tend to find is that there is hyperactivity in the anterior hippocampus, the limbic component of the hippocampus, in humans that correlates with the psychotic state of schizophrenia. Now, is the limbic hippocampus in the rat, which is the ventral hippocampus, hyperactive in the MAM animal? Well, if we just look at how fast the pyramidal neurons are firing, they are firing significantly faster. But of course, the hippocampus is an area that has a much more complex function than just how fast neurons are firing. Rhythmic activity is critically important for its function. What we find in the MAM rats is the normal slow oscillatory activity, the thing that drives up and down states in postsynaptic structures, like in the amygdala in the ventral striatum, is gone. Instead, they seem to be in a constant up state. But what we do get is an increase in the higher frequency rhythms. So what we do is we lose the gated low frequency rhythmic output and replace it more with a high frequency noise, if you will, that's coming out of this area. Now, how does this impact dopaminergic neuron activity? Well, if we look at our three parameters of dopaminergic neuron firing, number of neurons firing, firing rate, and firing pattern, what we find in the MAM animals is essentially all the neurons are firing all the time, without much of a change in average firing rate or firing pattern. Now, this is consistent with what's been done whenever you look at humans. Studies by Ollie Howes and Phil McGuire in London have shown that in both ultra-high-risk patients, patients showing attenuated psychotic signs, and schizophrenia patients, there is much greater fluorodopa uptake in the associative striatum compared to controls. Now, fluorodopa is taken up into active terminals. The more fluorodopa uptake, the more active terminals, the more dopaminergic neurons driving those terminals. And in fact, this also correlates with increased glutamate in the limbic hippocampus of schizophrenia patients. So there's a good consistency with what's seen clinically and what we're seeing preclinically. So what we again have is a hyperactive hippocampus, overdriving the ventral striatum, and removing palatal inhibition.
As you know, BTA has heterozygote neurons that receives inputs from different regions and sends their projections to the different regions. Which kind of neurons, for example, I think parabrachial and paraneural.
Right. So when we look, and I'll get into this a little bit later, but just as a preview, whenever we look at the medial-lateral distribution, it's primarily the lateral VTA that seems to show the greatest increase in number of neurons firing. And the lateral VTA tends to project more to the associative regions of the striatum. And the associative regions of the striatum are where you see the biggest amphetamine-induced raclopride displacement that correlates with psychosis.
So in this model, different parts?
So more lateral VTA, the VTA substantia border. Yeah.
Does this high frequency firing from the hippocampus disturb the
It does.
disturb the theta of the hippocampus high frequency firing?
You do see an increase in theta and higher frequency rhythms, not so much in gamma, at least not evoked gamma. Baseline gamma is high, but evoked gamma is actually suppressed. Mm-hmm.
We learned from the Michael and Anselmo that acetylcholine has a big role in regulating the activity of hippocampus.
What does?
Acetylcholine?
Yes.
So there's like we see kind of difference in this kind of hyperactivity that can be regulated by acetylcholine, or acetylcholine is somehow impaired in
There's a lot of literature about acetylcholine and schizophrenia. We've been looking at acetylcholine in these models, and we're getting some pretty dramatic differences. So we're looking at different alpha-7 drugs. Now, alpha-7 agonists are problematic because of the desensitization. But when we look at positive allosteric modulators that have alpha-7 receptors, whenever we look in control animals, we tend to get activation. When we look at the MAM animals, we get inhibition. And interestingly, whenever we looked at the septohippocampal pathway, it tends to activate the dopamine system in controls and inhibits the dopamine system in the MAM animals. So acetylcholine's having dramatically different effects in these two systems. And we're trying to figure out what it is, but we think it might have to do with pyramidal neuron, interneuron balance between what's happening with the inputs.
So I just missed out. What did you see when you use the alpha-7? So this is activated.
We tend to activate a number of neurons firing in the VTA of control animals and decrease it in the MAM animals. Now we know that this increase in number of neurons firing is mediated through the hippocampus, because if we inactivate the ventral hippocampus by injecting TTX, we bring the number of dopaminergic neurons firing back to control levels. And then, of course, we want to translate this to behavior. When we look at amphetamine-induced locomotion, we see much more locomotion in the MAM rats compared to the control rats. But if we inactivate the ventral hippocampus, that brings dopaminergic neuron firing back to baseline. We also bring the behavioral response to amphetamine back to baseline. So this all seems to be consistent with a hyperactive hippocampus, overdriving tonic dopaminergic neuron activity and making the system hyper-responsive.
So, other regions receiving inputs from the hippocampus aren't affected by this hyperactivity in the hippocampus?
The other regions the hippocampus projects to? Almost certainly. And the interesting thing is those regions tend to be involved in the other symptom states. And I'll talk about that a little later. Now, how does this increase in dopamine population activity affect the response of the dopamine system to behaviorally salient stimuli? Well, as I mentioned, one thing that this tends to be involved in is context dependency. The hippocampus, especially the hippocampus subiculum, is involved in context dependency, or the ability to regulate your response to stimuli depending on the context. And just like I talked about with benign contexts, when there's not a lot of neurons firing, you don't get a big dopamine response. If you're in an activating context, you get a big response. And you adjust the system depending on environmental contingencies. But the schizophrenic patient loses that. In the schizophrenic patient, all the neurons are firing because the hippocampus is overdriving the system all the time, which means they're always feel like they're in this threatening state where they have to pay attention to all stimuli. Now, whether a stimulus is salient or not, it causes a massive dopamine release. One thing that schizophrenic patients often complain about is being bombarded with stimuli with the inability to filter what's important and what's not important. In fact, there's an excellent book by Ellen Sachs called The Center Cannot Hold My Journey Through Madness, where she talks about her own experiences with schizophrenia. And one thing she describes is she says the filter that everybody has that tells them what's important and what's not important and what they should be paying attention to is broken so that everything floods in all the time. The other thing is it causes them to misinterpret stimuli. So for example, if you look outside of a window and see two complete strangers talking to each other, this is something you would just ignore because it's not relevant. But when a schizophrenic patient sees this, their dopamine system is inappropriately activated, which means their dopamine system is screaming at their brain, this is something really important to our survival that we have to pay attention to. And they start to confabulate, that these are aliens from another planet planning to abduct them, or this is the government spying on them, or all of these different things where they're trying to make sense of why this incongruent stimuli is causing such a big stress response in them.
Yeah, just a minute. What could be the relationship between these overshines with the allergies.
With-
Allergies. The over allergies.
Over alertness.
Over,
Alertness.
yeah.
What would be the relationship between these overshines with allergies?
So what they're doing is they're always having the dopamine system activated as if they are in a dangerous context where they have to pay attention to all stimuli. So in other words, you know how you feel when you made a wrong turn and you're in a really bad neighborhood and it's late at night and there's nobody around and all of a sudden this group of people starts walking towards you. Well, they feel like that all the time.
So just to make sure that I understand, so does this overactivation relate to therefore higher levels of tonic dopamine? But then, so to me, this idea of salient stimuli capturing your attention would actually be sort of more of a phasic response. Does that make sense? I don't know. So I'm trying to understand how the two kind of relate in terms of changes in baseline tonic dopamine levels and then how that might affect then sort of phasic. Because you said that phasic, there's no increase in the amount of phasic activity, it's only just an increase in the number of cells firing overall, right?
Right, there's an increase in the number of neurons firing. But what that means is now the system is going to be hyper-responsive to stimuli. So in the absence of stimuli, you just see an increase in tonic. But as soon as the stimulus comes in, you get a massive phasic burst.
Oh, so you do get an increase in, okay, I see.
Yeah, but it's evoked phasic.
I see, yeah.
But it's the most important reason of this hyperactivity in children.
I'll get to that.
Hyper-responsive to stimuli, it would mean that if you put some deprivation, that these guys would start feeling better, or there would be self-generated stimuli also, which would keep, them in
What would make it better?
sensory deprivation. If the stimuli is what is causing all this, then would in some way sensory deprivation lead to them not feeling so much? Because in schizophrenic patients, they also self-generate stimuli.
Of course, there's all these hallucinations and voices and things like that, but they're also very sensitive to stress, and stress tends to bring that out even more. So if you bring them into a very low stress environment, you can decrease the symptoms, but you're not going to get rid of them.
Sorry, just one very quick thing. So then if there is an increase in phasic response, because there's feedback from the accumbens to the VTA as well, right? So does that feedback have any, is that also dysregulated, or is there, does it essentially, because I know it's the hippocampus that's driving all of this, but that doesn't help, so to speak, but can't sort of try and reduce
There is going to be feedback inhibition, but the problem is the hippocampus is driving the steady state into an activated state. So even though there may be some feedback inhibition, the initial drive is going to still be much stronger.
Does ketamine has any positive effect in schizophrenia?
Ketamine? Yeah, it makes it a whole lot worse.
It's an intermediate agonist. It's an antagonist, right? Of NMDA receptors.
It is, but when you're giving it systemically, what you tend to do is to get a big activation of the thalamo-hippocampal pathway, which causes, and it also tends to block glutamatergic drive of parvalbumin interneurons. And the combination of those means that you're going to end up with an overactive hippocampus.
I was thinking more on effect, the dopaminergic drive.
Yeah. Unfortunately, whenever you give it systemically, it's going to have its biggest effect where you have the biggest transmission, and the biggest transmission is going to be in this hyperactive pathway.
A lot of your results you're thinking about in terms of the hippocampal striatal pathway in BTA. There's also a strong hippocampal lateral septum pathway in BTA. Do you think that's mediating a similar set of changes or doing something different?
So we've looked at the different pathways, and that pathway has an impact, but it's not nearly as great of an impact as this pathway. So I know in your papers you show that this has an impact, but if you remove that pathway, it doesn't really decrease it that much. Whereas if we interrupt this at any one of these stages, it completely changes the responsivity. So especially
in your man model in particular.
in the controls and in the MAM models, but especially in the MAM model. Yeah.
So how could we reconcile the fact that typical antipsychotics work as antagonists on the D2s, and think that you're thereby inhibiting the autoreceptors and increasing dopamine release? It's almost driving this hyperactive state into a more hyperactive one. You're almost like heightening the tone of dopamine. So,
Okay.
so it's in contradictory ways.
So if you remember what we said here about supersensitivity and spare receptors, the D2 antagonist is not going to discriminate between autoreceptors and postsynaptic receptors because they're the same receptors, except that there are spare receptors in the autoreceptor, which means when you start blocking the autoreceptor, you're still going to have a number of unblocked receptors that's higher than the number of unblocked receptors postsynaptically. So the increase in dopamine release by autoreceptor stimulation isn't going to be nearly as much as the attenuation from postsynaptic blockade. But the interesting thing is that postsynaptic blockade is not enough on its own, because the thing about postsynaptic blockade is you can compensate for it. You can increase receptor number, you can increase tyrosine hydroxylation. But what we find is if we give antipsychotic drugs repeatedly, you actually get this overactivation of the dopaminergic system to such an extent that it causes depolarization block. It essentially shuts them off. And that brings the number of neurons firing back down to normal. Interestingly, that happens faster in the MAM rat than it does in the control rat. Control rat, it takes 3 weeks. MAM rat, it takes a single dose. And that correlates better with the therapeutic response and with the fact that antipsychotic drugs have a stronger and faster effect the more psychotic the patient is. And in fact, Ollie House and Phil McGuire have shown that if you don't have a high level of fluorodopa uptake, they tend to be treatment resistant. So overactivated dopamine systems correspond to stronger activation by antipsychotic drugs, correspond to better therapeutic action.
Does that help to explain the off-side effects as well? A lot of the results of antipsychotic treatments are extrapyramidal symptoms, dyskinesias of various sorts.
That has to do with first generation, second generation drugs. So the second generation drugs don't really have the EPS. The EPS you see with more first generation drugs like haloperidol, things like that. Clozapine, Seroquel, you don't tend to have EPS. The EPS tends to correlate with the drugs having a stronger action on the substantia nigra. So whenever you get the polarization block in the substantia nigra, you get EPS. You give an anticholinergic, brings them out of block, and you get normal behavior. Mm-hmm.
So the amphetamine-induced locomotor activity,
The
amphetamine-induced increase in locomotor activity, is that still through the VTA dopaminergic system or through the substantia nigra dopaminergic system?
Amphetamine has a much stronger effect than the VTA system. And we think it's part of this reuptake blockade that's accentuating the phasic response that's causing that. Okay. So basically, when the hippocampus is hyperactive, the dopamine system is hyperresponsive to stimuli, and we think this is why you get these overinterpretation of events. Now, as I mentioned, the ventral hippocampus is involved in context dependency. And schizophrenics are notoriously bad at context dependency. And in fact, when we look at these rats, we see the same thing. So here we have two different contexts. Context A, that's defined by a tone, a red light, and an odor of glacial acetic acid. Context B, that's a different shape, that doesn't have a tone, that has a green light, and has a tone of ammonia. We put them in the first context and expose them to foot shock. We then give them a break and put the rats either in the exposed context or the non-exposed context. Now, just like you might expect, rats, normal rats that are in the exposed context freeze, and in the non-exposed context don't freeze. MAM rats, you don't see a difference. And this is exactly what you see in schizophrenic patients that are put into different environments, one where they've experienced punishing stimuli, one neutral stimuli, and you rate their level of uneasiness. Normal patients are very uneasy in the punished environment, no response in the neutral environment. Schizophrenic patients don't differentiate between the two. So they lose this context dependency because you lose the ability to modulate hippocampus depending on context. So these data show that the MAM model of schizophrenia, ventral hippocampal hyperactivity, leads to a hyperresponsive dopamine system that we think underlies psychosis. Why is the hippocampus hyperactive? That was something that somebody asked earlier. The most robust finding that's...
Would you expect in the MAM rats to see freezing in both contexts? You can't really respond in a sort of normal manner.
That's interesting. Why aren't both maximal instead of both minimal? And I think it's basically because since they've experienced this over their entire lives, they've accommodated to it. But what they've lost is the ability to discriminate. One of the most robust findings that's been reported in schizophrenia patients post-mortem is a substantial loss of parvalbumin. Parvalbumin is contained in GABAergic neurons, especially the ones that synapse perisynaptically. And you see it in both frontal cortex and in hippocampus. And you see about a 30% decrease. Now, if you know anything about post-mortem studies, a 30% decrease in a marker is enormous. So this has been reported in hippocampus in schizophrenia patients. We see the same thing in our MAM rats, a decrease in medial prefrontal cortex and ventral hippocampus parvalbumin. Now, the thing about parvalbumin is metabolically expensive to make. You don't make it unless you have to. So if you're not being activated, you don't make parvalbumin. If you're dead, you don't make parvalbumin. So are the neurons dead or are they not active? Well, we actually looked at this, and we took a hint from people who did work looking at temporal lobe epilepsy, where you also see parvalbumin loss, and interestingly, psychotic features. But what they found is that you can stain parvalbumin neurons for substance P receptors that are constitutively expressed independent of activity states. So what we did is co-stain for substance P receptors in parvalbumin. And what we found is when the rats are very young, we tend to see a decrease in parvalbumin content, but not in substance P receptors. But when they reach adulthood, we see a decrease in both markers. So initially there's a loss of parvalbumin, but in adulthood, the neurons are dead. This is in the hippocampus. Interestingly, David Lewis has said in the frontal cortex, you just see a decrease in parvalbumin content per neuron without neuron loss. And that's what we also see in the frontal cortex, a decrease in parvalbumin neuron markers. And that probably has to do with developmental staging that I'll get to a little bit later. Now, what is so important about parvalbumin interneurons? Well, when a neuron fires, calcium comes into the neuron. Calcium activates a calcium-activated potassium conductance that causes a hyperpolarization that delays the next spike. The faster you sequester calcium, the faster the neuron can fire. Parvalbumin is the highest affinity of the calcium-binding proteins, which means parvalbumin neurons can fire like gangbusters. They can fire at 40 hertz, which is enormous. So they're really important for high-frequency gamma rhythms that are 40 hertz or 80 hertz that are known to be important in stimulus recognition and processing. And interestingly, whenever you look at schizophrenia patients, what you find is though all baseline gamma may be activated, evoked gamma is lost. So we actually tested this in the MAM rats by putting field potential electrodes either in ventral hippocampus and medial prefrontal cortex, exposed animals to either a foot shock paired with a tone or a non-paired or neutral tone, and then used fast Fourier transform to break down the local field potentials into their frequency components. And what we found is that in normal rats, the tone exposed to the foot shock activates gamma in the medial prefrontal cortex and ventral hippocampus, but there's no activation in the MAM rats. So just like in the schizophrenia patients, they lose the stimulus-evoked gamma rhythmicity in the same regions where the parvalbumin neurons are lost. And this is just to show you graphically about this regionally selective decrease. So what I've been talking about, again, is ventral hippocampus overdriving the dopamine system and how it relates to psychosis. But as I mentioned, schizophrenia is more than just psychosis. There's also the negative symptoms and the thought disorder. The thing is, if you leash the ventral hippocampus, you can get all kinds of compensatory processes coming in. But when this area is hyperactive and dysrhythmic, you can't compensate for that. Ventral hippocampus also has projections to the amygdala, which through the cingulate and orbital frontal cortex is involved in emotional regulation. If you disrupt this, you get the negative symptoms. It also has projections to the prefrontal cortex, where it's involved in cognition. We know about theta-modulated gamma rhythms that are so important for prefrontal function. So when this area is hyperactive and dysrhythmic, it has the potential of dragging the whole circuit down. Treating here fixes the psychosis. It doesn't fix this. And in fact, antipsychotic drugs are terrible at cognitive and negative symptoms. So evidence suggests that both in schizophrenia and the MAM model, there's hyperactivity in the ventral hippocampus, possibly because of decreased interneuron function. And if we inactivate the ventral hippocampus, in the MAM model, we can restore normal dopamine function. Now, how do we restore normal dopamine system function in schizophrenia? Well, what we have are sites of deficit that we think are in the hippocampus. And this is what we think leads to the overactive dopamine system. But where do we treat schizophrenia? We block dopamine here. 1, 2, 3, 4, 5 synapses downstream from where we think the deficits are, which is probably why these antipsychotic drugs are such, although they work, are such lousy drugs. Patients don't like to take them. There are still so many residual symptoms. A better plan would be if we could actually work at the site of deficit. And that's what a number of people are working on now. I don't know if you know how antipsychotic drugs were developed, but they weren't discovered to treat schizophrenia. They were discovered trying to treat hay fever. It basically started out with a drug called promazine, which is a prescription antihistamine. But like all antihistamines, it's horribly sedating. So what they did is they started manipulating the molecule, and they chlorinated it. And they were real disappointed because it was still sedating, and now it didn't have any antihistamine properties. But this French surgeon, Labourite, found that he could give it to patients undergoing surgery and stabilize them for very long periods of time so that they could have these extended surgeries. And he coined the term neuroleptic for neural stabilizer. Well, at this time, there were no treatments for psychiatric disorders. You kept people sort of hidden in your basement until you couldn't deal with them anymore, and then stuck them in the asylum where they spent the rest of their lives. Well, DeLay and Deniquet heard about a neural stabilizer and thought, what a great drug to give to patients. So they gave it to everybody. But the schizophrenics all started to get better. They started to move more, they started to get motivation, they started to lose their hallucinations. This was actually the first antipsychotic drug, chlorpromazine or thorazine, which was actually discovered to be effective 10 years before dopamine was discovered to be an independent transmitter in the brain. So it was all by serendipity, something that drug trials would never let us do today. So a better approach would be treating at the site of pathology. And this is something that we and a number of pharmaceutical companies have been working on, and we have some good hints at that, and I can fill you in on some of those details later on if you're interested. Let me go past this because I don't think I have time to talk about this. So in schizophrenia in the MAM model, interneuron dysfunction may be a primary driver of the symptomatology. And if we can restore interneuron function, we might have a better therapeutic response. The problem is schizophrenia is bad for the brain. The longer you have untreated psychosis, the worse the prognosis. A better approach would be as if we could prevent schizophrenia to begin with. Now, how can we do this? Well, one thing that's been correlated a lot with schizophrenia is stress. We know that stressful stimuli will exacerbate positive psychotic symptoms in schizophrenia patients. And even normal individuals under very stressful conditions can start to show some attenuated transient psychotic signs. But it also may play a role in the etiology. Some studies that were done by Eve Johnstone and Robin Murray looked at children that were at genetic risk for schizophrenia based on family history and did a bunch of psychometric tests and then followed them to see who converted and who didn't. And what they found are the children that were hyper-responsive to stress early in life tended to convert to schizophrenia later in life. And the ones that didn't show this didn't convert. And the thing about stress is known to cause a loss of parvalbumin interneurons in the hippocampus, which are the same neurons lost in schizophrenia patients in the MAM rats. The premise is in normal individuals, we have things that help us to deal with stress, especially the medial prefrontal cortex. But if this area isn't working optimally, these deficits we think can initiate a cascade of events that magnify the effects of stress, lead to interneuron loss and dysfunction in the adult. So if you stress an animal, one thing that you see is a big activation of the mesocortical dopamine system. The mesocortical dopamine system activates the medial prefrontal cortex and then feeds back and inhibits the stress response. In fact, if you disrupt this, you get a much bigger stress response. But what about in the schizophrenia patient? Well, what we think is that there's a deficit early on, either in dopamine modulation of the prefrontal cortex or in prefrontal cortical function. Because of this, there's a dysregulation of the dopamine system that's a stressor. The medial prefrontal cortex, especially the prelimbic cortex, is known to inhibit amygdala response to stress, which means you lose this inhibitory pathway, which magnifies the effects of stress. You also lose frontal cortical down modulation of the locus coeruleus, which can lead to more stress, which leads to hypothalamic and glucocorticoid release, hippocampal damage, and so on, in this positive feedback loop where stress generates more stress, generates damage, generates more stress. Now, if this were true, you would expect that the MAM rats would be more sensitive to stressors very early on in life, just like the schizophrenia patients. Well, if we look at our MAM rats very early on, postnatal day 22, puberty in male rats is about postnatal day 40, so this is pretty early on. And we do foot shock evoked vocalizations. What we find, especially early on in life, are the MAM rats show much more and intense foot shock vocalizations than control rats. I mean, this ultrasonic vocalization is really an interesting phenomenon. You've often heard stress-induced freezing, where the rats sort of splay out and freeze. They're not being passive. When they're doing that, they're going, ah, ultrasonically. Never stress a rat in a rat colony, because all the other rats are going to hear them screaming. And what we find, again, is that the MAM rats are showing much more of this ultrasonic vocalization. When we look at elevated plus maze, this is where we have a plus-shaped maze where two arms are enclosed and two arms are open, and it's suspended on a platform. Normal rats will explore all the arms because they're curious. The more anxious a rat is, the more they hide in the closed arms. MAM rats hide in the closed arms way more than the control rats. And finally, when we look at the amygdala, an area that's known to be associated with anxiety and stress, even again, before puberty, the amygdala, basolateral amygdala, is firing significantly faster than what you see in control rats. So all these measures of stress and anxiety are off the roof before they're showing any of the hyperactive dopaminergic states. Now, if stress plays a role in this developmental etiology of schizophrenia, This suggests if we can intervene early enough and control stress, we might be able to prevent the transition to schizophrenia.
So how do we predict that early on? I mean, why do we predict the vulnerability to stress at an early age in humans? Is there a category for us to detect, like, to be a certain threshold, like, you're more likely to develop psychosis and later in life? Do we have that?
So what you would have to do is to do a two-stage thing. First, you look at family history, because schizophrenia tends to run in families. Then you take the children and do simple stress measures. So you look at galvanic skin response, which is a very non-invasive thing, and then look at social interaction, which can be very stressful, like speaking to a group, like me. My GSR is off the roof. And look at the children that are showing these responsiveness to stress.
You can also use saliva cortisol.
Yeah, that's another way. And then what you want to do is just get them into an environment where you can teach them how to deal with stress, like with cognitive behavioral therapy. Yeah.
Just, what about the very gradual age of the rat and so on? What about the relationships with the growth hormones and the hormones classically?
There doesn't seem to be a good correlation with the age of onset of schizophrenia with those parameters. But what I'll show you is something that I think is more relevant. Now, as I mentioned, if stress plays a developmental role in schizophrenia, we should be able to intervene early. And what we did is to take MAM rats and give them the anti-anxiety agent diazepam or Valium right before puberty. And what we got is a dose of diazepam that normalized behavior in the elevated plus maze and normalized amygdala firing in this prepubertal state. So something that was just enough to reverse anxiety. We then gave the rats either gestational MAM or saline, gave them 10 days of diazepam, waited until they reached adulthood, and then start to look at different parameters. 10 days of diazepam right before puberty was enough to decrease the parvalbumin neuron loss and prevent the activation of dopaminergic neuron firing. So if we gave them vehicle for 10 days, we still got our activation in the number of dopaminergic neurons. If we gave them diazepam for 10 days, withdrew it and waited till they reach adulthood, dopaminergic neuron firing is normal. And we also normalized the behavioral response to amphetamine. So we prevented the emergence of the hyperdopaminergic state.
Did you just say that if stress is such an important factor for this schizophrenic feature, then didn't you say that women are more sensitive to stress? Why are females more protected from schizophrenia?
It's a really interesting question, and it has to do with the way stress affects females early in life. And it affects females early in life very differently than it affects males. So if you take, and I might get this wrong, I have it just written in my grant, but it didn't get funded, so. If you stress male and female rats before puberty, and then look at them as adults, female rats tend to show more anxiety, but male rats tend to show more cognitive deficits. So that the way the stress affects the system early in life is different. And that probably has to do with this kind of vulnerability. Now, this is interesting because, as I mentioned, converging evidence from clinical and basic science suggests the inability to regulate stress can lead to these pathological changes. But the fact that we can circumvent MAM by controlling stress suggests MAM doesn't cause schizophrenia. MAM causes hyperresponsivity to stress, and hyperresponsivity to stress causes schizophrenia. And I think this is why so many people who are looking at a genetic basis for schizophrenia might be looking at the wrong thing. Because there's not a lot of evidence that genes cause schizophrenia. There are associations, but there are 108 genes associated with schizophrenia. Even identical twins only show a 50% concordance for schizophrenia, even though their genetics are identical. And a third to a half of schizophrenics have no family history of schizophrenia. What I'm suggesting is it might actually be the genes that are doing the same thing, changing the response to the environment, and it's the genes potentiating the environment that leads to the pathology. Did you have a question?
Yeah, actually, so how does this fit into, because one of the biggest environmental risk factors is stress of the mother in the second and third trimester, and also like inflammation.
So I think that the inflammation and stress of the mother are some of the things that predispose the individual to greater stress responses around puberty. So as I mentioned, it's a gestational disruption, either genetic, maternal conditions, things like that, that somehow cause the animal to be hyper-responsive to stress around puberty, and it's the addition of the stress around puberty that then leads to the pathology. And I'll give you some evidence for that later.
Given this important role of stress in developing schizophrenia, do demographics indicate that the propensity of schizophrenia is greater in more stressful environments, in poorer countries, where just the general stress level is much higher than in developed Western?
No, absolutely. There are studies that have shown that Jamaicans that moved to the UK, their children have a 17-fold higher incidence of schizophrenia than the general population. Now, you might think you'd have to be crazy to move from Jamaica to the UK to begin with, but it's the social stress that the children have when they try to integrate into this foreign population, and social stressors are very, very bad.
Is this even if they don't move, just in those populations that have this chronic stress because of the low economic status?
Well, it's actually the opposite, because if you look at the barrios surrounding Sao Paulo, Brazil, people there are very poor. But because they're poor, they don't move. They have a great extended family ties and support systems. And what they find is the incidence of major psychiatric disorders is significantly less in that population than it is in wealthier populations that move around a lot. And Robin Murray showed that probably one of the greatest risk factors for schizophrenia are the number of times a family moves when the child is between 10 and 15 years of age. Because every time they move, they have to reestablish all new social bonds, which is extremely stressful.
Seems like malnutrition, stresses like that should have a...
Oh yeah, they do.
It raises an interesting point, because we know in schizophrenia, urbanicity, and alienation are the two key stressors of the human data. But is this asking us questions about the nature of stress? We refer to stress as a monolithic thing. You might expect increased stress at this time to produce more depression. I'm sure you do get a bit more depression, but...
And I'll give you an answer to that in a little while.
So I guess the issue is, does stress come in different flavours? And are these flavours conveyed by different patterns of activity in these chemical modulatory and endocrine systems?
The critical thing is the stage of development in which the stress occurs. And I'll show you why in a little while.
Clarification. You did show that given the diazepam to advanced animals reduce the loss of pyramidal neurons, correct?
Right.
Sorry, actually that,
Yeah.
So in man animals, the amygdala also had increased activity.
Yes.
Right? But from the previous circuit, we know that the amygdala actually inhibits dopaminergic tone. So then, how is it that although there is increased activity actually in the amygdala, in these mammals there is also increased dopaminergic hyperactivity?
So the amygdala has a number of different projections. It projects to the ventral pallidum to control dopamine. It also projects to the hippocampus. And whether it's selective for the hippocampal projecting neurons or whether the hippocampal projecting system predominates, we're not sure. Because one thing you have in the normal animal is the hippocampal amygdala balance, that in depression the hippocampal pathway drops out. Well, it doesn't drop out here, it's actually hyperactive. Yeah.
Is giving diazepam to adult schizophrenic patients, is that sufficient to compensate for the loss of TB in the hippocampus, or is it uncompensatable?
No. There are some schizophrenic patients that do very well on diazepam. In fact, every schizophrenic patient is given diazepam because they're all anxious. And I'd be surprised if you found one that didn't have it. I mean, they're all on 10 drugs. The problem with diazepam is it's hitting the wrong receptors. Diazepam is anti-anxiety by hitting primarily alpha-1, alpha-2 receptors. But the hippocampus is loaded with alpha-5 receptors. And we can actually normalize this condition by giving a GABA-A alpha-5 positive allosteric modulator that beautifully restores the system to normal in the adult. Yeah.
How long do you expect that the diazepam treatment of children should be? And would it have do you think it could create a dependency if they have to take it for years?
No, I don't think we should be putting diazepam in cornflakes or anything like that. The thing is with humans, you can do cognitive behavioral therapy. You can teach them how to deal with stress. You can't do that with rats very easily. But you can change their environmental contingencies, which is something that we're trying to do. And actually, Pat McGorry in Australia has been doing some work where he's been getting at-risk children and getting them into highly social interactive environments to try to prevent the emergence of pathologies later in life. But of course, this is a long-term project.
And also difficult for children with parents with schizophrenia because they're normally in the social class.
Exactly. Yeah. Go ahead.
You mentioned that the projection to cortex and amygdala in the mammal try to explain to positive, negative and cognitive centers. Is there any projection from mental hippocampus to somewhere else that may explain to the positive symptoms as well? Maybe should be cognitive symptoms.
Yeah, we think it's the hippocampal projections to the ventral striatum, ventral pallidum, dopaminergic system that causes the positive symptoms by making the dopamine system hyper-responsive. Yeah.
Do you think that the control of production or action of stress hormone can also be part of early treatment?
That's a double-edged sword. Because whenever we look at MAM animals, they actually show blunted corticosterone responses. If you look at the literature, although corticosterone is released to stress, corticosterone or cortisol in humans is necessary for homeostatic compensations for stress. In fact, there were studies that were done by Zohar in Israel showing that people with PTSD have a blunted cortisol response. And if he gives them cortisol, he can actually treat the PTSD. So not having a cortisol response is worse.
So maybe improve it to give it to them.
Could be, yeah. Okay. So, okay, so this suggests that if stress plays a big role, if we stress a rat enough, especially prepubertally, we should be able to get parvalbumin loss in a psychotic-like state. So what we did is to take rats right before puberty and give them stress exposure. We either gave them 3 sessions of restraint stress for 1 hour or daily foot shocks, 25 shocks each day for 10 days, or combined foot shock and restraint, and then looked at different tests after they reached adulthood. Now, interestingly, whenever we gave them these restraint stress, the animals were way more anxious. Not as much with foot shock, but the combined foot shock and restraint, as adults, they were more anxious. When we looked at novel object recognition, a type of cognitive test, interestingly, the restraint stress didn't have as much of an impact in novel object recognition. But foot shock pre-pubertally really disrupted novel object recognition, as did the combined foot shock and restraint. But when we looked at the dopaminergic system, only the combined prepubertal restraint and foot shock gave a significantly greater amphetamine-induced locomotor response and a significant increase in the number of dopaminergic neurons firing. Now, interestingly, when we look at which dopaminergic neurons are activated, it's exclusively the ones in the lateral VTA, the ones that projected the associative striatum, which is the area where you see the biggest raclopride displacement in schizophrenia patients. What about if we do this in adults? We give adults this stress exposure and then wait the same period of time and start looking at these parameters? Nothing. No, we do see some body weight gain, but they catch up. No differences in anxiety, novel object recognition, amphetamine response, or VTA dopamine activity. So what's special about this early period that can lead to schizophrenia? Well, first, let me back up. Why is it that we have this hyperresponsivity to stress? So stress can lead to activation of the basolateral amygdala. It also leads to oxidative stress. And oxidative stress damages parvalbumin neurons. In fact, because these neurons fire so fast, they're really susceptible to disruption by overdrive. So you lose parvalbumin neurons, activate the ventral hippocampus, and so on. One thing that really controls basolateral amygdala responsivity to stress is the medial prefrontal cortex. In this case, the prelimbic prefrontal cortex. The prelimbic prefrontal cortex projects the GABA neurons within the basolateral amygdala and suppresses amygdala responses to stressors. What if this area wasn't working? So what we did is to go very early on, postnatal day 25, and lesion the prelimbic prefrontal cortex. We then looked at the animals, again, around this postnatal day 30 to 40, before puberty, and exposed them to stress. But now we did just foot shock, something that was inadequate to cause the pathology before. What we find is the medial prefrontal cortical lesion at postnatal day 25 was enough to make them anxious already at the prepubertal time point. So lesioning the prefrontal cortex made them look just like mam rats with respect to anxiety and stress responses. Now, when we go in and give them just foot shock, those 10 exposures of foot shock, during the prepubertal period, we get an increased amphetamine response and an increased number of dopaminergic neurons firing. So we potentiated the deleterious effects of a more minor stress to make it look like the extreme stress. And now we get a big activation of dopaminergic neuron firing, and it actually looks more like the mam rats, where we get it more across medial-lateral extent than just exclusively in the lateral area.
Can you do something like immunization, where you can expose them to stressors that they can't control, and then you expose them to stressors which are awareness, and you see less of an effect on the
That's a really interesting idea, and we know that it works well in adults, but whether it works when the prefrontal cortex isn't fully developed is another matter. So that's something that would have to be explored. Doing okay? Now,
have you tried several intensity of foot shock or just high intensity?
Just the one high intensity. These are long experiments. So can we make adults more vulnerable by reopening the critical period? Now, what is it about parvalbumin neurons that makes them so sensitive? Well, parvalbumin neurons in the prepubertal state are in a very plastic condition. There are a lot of glutamatergic synapses that are being made and broken as the animal learns to adjust to the environment and environmental contingencies. After puberty, but while they're very plastic, they're also very sensitive to oxidative stress and metabolic stressors. After puberty, they get these polyglycan perineuronal nets around them that locks in the plasticity. So they lose the plasticity, but they're protected. So now they're not as susceptible to oxidative stress. And this is called the critical period of development. This is something that was studied very well by Takao Hensch in the frontal cortex, looking at ocular dominance columns. Really fascinating literature. So the critical period is opened with the very early on in life, but especially with GABAergic processes maturing, but then it's closed by a number of things: perineuronal net formation, links with cholinergic system, and a number of other things. But you can actually reopen the perineuronal net in the adults if you give an HDAC inhibitor. So what we did is to use valproic acid, which is known to be an HDAC inhibitor. We started giving it to adult rats, young adults at postnatal day 60, and then gave them the combined stressors between day 65 and 75. And what we found is if we give them the combined stressors to a normal rat and then measure just a week or two later, we get an inhibition in the number of dopaminergic neurons firing, something that looks more like depression. But if we reopen the critical period with valproic acid, now the same stressors increase the number of dopaminergic neurons firing to look more like schizophrenia. Now this is interesting because schizophrenia and depression run in the same families. And it might be that they're both hyperresponsive to stress, but it depends when that stressor occurs. If it occurs early in life, you get schizophrenia. If it occurs later in life, when the parvalbumin neurons are protected, you get depression. If you reopen the critical period, you can get this as well. Now this raises some interesting questions because valproic acid is a mood stabilizer. And other things that open the critical period are things like tricyclic antidepressants. And tricyclic antidepressants have very variable effects. Some people respond well, some people don't. Maybe the environment that they're given these drugs are critically dependent on the outcome. Did you have a question?
You mean that when a natural brain is under stress, almost susceptible to get schizophrenia. So
Exactly.
What's your justification for patients who are not under stress in adolescent or childhood period?
So it's probably a combination of genetic predisposition and environmental factors. So if you have no genetic predisposition, huge amount of stress, you can get schizophrenia. If you have a small amount of genetic predisposition, lesser levels of stress, you get schizophrenia. Very high genetic predisposition, moderate levels of stress, get schizophrenia. Our MAM rats, just housing them in wire cages is enough to get the pathology. We were interacting with a drug company that treats rats way nicer than we do, and they use the solid bottom cages and controlled environments and all this stuff, and they weren't getting the phenotype until they put them in wire bottom cages, then they got the phenotype. So really those conditions right before puberty are critical about whether you get this transition or not.
And does it work by this mechanism?
That's what we think, but that hasn't been tested yet. What you're seeing is everything we have. In fact, all this stuff we're still trying to finish up. So therefore, the deleterious impacts of stress combined with factors predisposing an individual to the impact of stress, we think underlie susceptibility to schizophrenia. And it's critically dependent on the timing of the stress. If the stress occurs during adolescence, you can get parvalbumin loss and emergence of schizophrenia. If exposure occurs in adulthood, when the parvalbumin neurons are protected, you're more likely to get depression. And the increased response to stress may be mediated by a failure of the medial prefrontal cortex to limit the impact of stress exposure. And we think that this is where the gestational factors are involved. The maternal stress, the immune activation, genetic predisposition, those kinds of things. And if we can identify susceptible individuals and intervene early enough, we might be able to prevent these deleterious effects and transition to schizophrenia. Yeah.
It's possible there is some epigenetic in schizophrenia, meaning like
it certainly is. I mean, the genetics of psychiatric disorders is really a complex type of thing. It's not like Huntington's, where you have the gene, you get the disorder. It's an interaction of a large number of genes with minor impact. I've heard a great description that you can describe susceptibility as a bucket of bad luck. And it doesn't make any difference what you put into the bucket. It's genetic predisposition, maternal stress, prepubertal stress, but as soon as you fill up the bucket, you get the disease. And that's what we're seeing here. You know, you need MAM plus minor stress, you need major stress by itself, you need prefrontal cortical disruption and minor levels of stress. You just need these interacting variables. And once you pass the threshold, you get the parvalbumin loss and the emergence of pathology.
I think we have to go for lunch.
Okay.
So thank you.